presenters to come up and excite us and wake us up from our post-lunch slumber. So I could ask them to come on up. It's Nida, Shahid, Bedra, Sharif, Zaved, Waise, Tatsuaki, Watanabe, and Whyte. And then I just want to remind everybody that we have a hard end at 4:10 today. And I really want to come back and congratulate the poster award recipients. And so I'm going to just get all the judges ready. You've got your pink sheets. And we're going to have about 20-25 minutes after these flash presenters for our first set of posters before our next set of talks. And then there's a second round of posters. So come on up. We'll start with Nida.
Hi everyone, my name's Nida. I am a research associate at Theta Collaborative and a PhD student at IHPME. I am really excited to talk about my project, and I invite you to come see me at Poster 60 to talk about a very layman's overview of the different various applications of neural nets in healthcare decision making. In addition to the study findings related to where these studies came from and what they tackled to do, or aim to do, and the challenges identified by the authors, I'm excited to talk about the review process. We were recently awarded a prize by U of T Libraries for our search syntax, our search strategy, and the use of sources. And yeah, I'm really excited to talk about it, so I hope you come and see me at Poster 60. Thank you.
Bedra?
Good afternoon. We have found that PAX2 mutations cause 5% of adult-onset FSGS, one of the leading causes of kidney failure. By virtue of PAX2's role in differentiating progenitors, people hypothesize that PAX2 expressing cells retain a multipotent ability in the adult kidney. We hypothesize that PAX2 mutations impair the kidney's ability to repair. In a PAX2 mouse model, we show that these mice have smaller kidneys, normal kidney function, and normal lifespan. However, when we deliver a glomerular toxin called adriamycin, we see accentuated kidney disease. We also see cell death in PAX2 expressing cells, which we speculate reflects a failed effort of these cells to fulfill their regenerative role. Our work is important because understanding kidney regeneration has implications for a broad range of kidney diseases. Thank you.
Zaved?
Good afternoon, everyone. I am Zaved from Dr. Tony Lam Lab. I am as a postdoctoral fellow. I am now investigating the new drug targets to treat diabetes. We all know diabetes is the most common complicated disease and metabolic disease where metformin is the first-line therapeutics. Recently, it has been shown that metformin alters our gut microbiota and also affect our gut hormones, and finally affect the modulate gut-brain axis to regulate whole body glucose homeostasis. However, the exact molecular target of metformin at the level of the gut is unknown. And here in our study, we are showing that metformin inhibits the metformin inhibits the mechanistic target of rapamycin that is called mTOR at the level of the intestine to regulate whole body glucose homeostasis. We further confirm the role of mTOR at the level of the intestine by including both molecular and chemical methods. If you are interested to know more about our research, please visit poster number 4. Thank you.
Tatsuaki.
Good afternoon. My name is Tatsuaki Watanabe, a research fellow at Thoracic Surgery Laboratory. Chronic rejection after lung transplantation is a primary obstacle for long-term survival after lung transplantation. Antidonor immune response is a major reason for this. In addition, ischemia reperfusion injury at the time of lung transplantation and airway inflammation caused by pathogens, pollutants after lung transplantation are thought to augment lung immune response and then result in chronic rejection. We developed a mouse lung transplantation model with chronic rejection phenotype depending on autoimmune response and ischemic reperfusion injury. We observed that tertiary lymphoid organ, TLO formation, is increased in allografts experiencing ischemic reperfusion injury. Interestingly, with the treatment of B-cell depletion antibody, we observed reduced number of TLO formation and attenuation of chronic rejection. Our observation suggests that lung transplant recipient experiencing ischemic reperfusion injury, lung transplant recipient experiencing ischemic reperfusion injury, B cells have an important role in the development of chronic rejection. My poster number is 68. I'm happy to talk about this experiment. Thank you very much for your attention.
And come on up, Rachel.
Good afternoon, everyone. My name is Rachel Whyte, and I am a first-year master's student at the University of Toronto. The study that I am presenting today is part of the AMBERS Osteoarthritis and Osteoporosis Research Program. The overarching hypothesis of this research program is that knee osteoarthritis in postmenopausal women is actually caused by osteoporosis, especially in those who are non-overweight. In fact, previous analysis have shown that approximately 30% of postmenopausal women with knee osteoarthritis also have osteoporosis. In order to further examine this, we created a cross-sectional study at baseline that compares postmenopausal women with both knee osteoarthritis and osteoporosis to those who just have knee osteoarthritis and those who just have osteoporosis. As you can see on the table in the slide, we concluded that postmenopausal women with both diseases were not distinct from the group who just had osteoporosis. However, they were distinct from the group who had just knee osteoarthritis. So if you would like to learn more, feel free to stop by poster number 9. Thank you.
So with those great flash presentations, how can you not go to those posters? So again, judges, it's the odd numbered posters where the presenters are expected to be there over the next 20, 25 minutes. And there's prizes that'll be announced at the end. So I invite everyone to go and view the odd number posters, and then we'll do the even ones in our next session. Please be back at 1:30 or 1:35 for our final oral sessions. Thank you.